Hi, everyone. My name is Mark Stukel. I am a visiting PhD student from Chris Simon's lab at the University of Connecticut, and I have been visiting CCE at the University of Auckland for the past year on a Fulbright Fellowship. Today, I'll be talking to you about one of the projects I have been working on in my Fulbright Fellowship, which involves phylogenetic network methods and New Zealand cicadas. So one of the many processes that has generated biodiversity in Earth's history is rapid species radiations, in which a single species has diversified into many different lineages over in a short period of time, usually as a result of encountering a bunch of new environments. Another evolutionary process that can occur during a species radiation is hybridization. And hybridization may occur during a species radiation through several different processes, such as speciation with gene flow or secondary contact between diversifying lineages that have been separated geographically. And in some cases, hybridization may influence the dynamics of a species radiation by allowing adaptive genes to pass across species boundaries. That brings me to my study system, which is New Zealand cicadas. There are around 50 species of cicadas in New Zealand, currently classified in 5 genera. Of these genera, 3 of them, Kikihia, Maoricicada, and Rhodopsalta, contain the vast majority of these species, and they all descend from a common ancestor that arrived in New Zealand around 11 million years ago. In particular, I'll be focusing on the genera Kikihia and Maoricicada because they contain most of the species in this species radiation. Kikihia has around 30 species in New Zealand, about half of which are currently undescribed. And Kikihia species are distributed primarily low to mid-elevation with some subalpine species. And their habitat varies from grasslands to shrublands to forests on both North and South Island. While Maoricicada has around 15 species, and they are distributed low to mid-elevation all the way to the high alpine, with most of the species diversity up in the mountains. And many Maoricicada species live on rocky slopes well above the tree line, with some making it all the way up to the snow line, which is very impressive for an insect. As part of previous work on New Zealand cicadas, I was building trees using a phylogenomic dataset of around 500 nuclear genes and mitochondrial genomes. And in the process of building phylogenetic trees for the species radiation, I uncovered large amounts of mitonuclear discordance in both Kikihia and Maoricicada, all of which could potentially be explained by ancient hybridization. So first of all, for example, in Kikihia, on the left, we have an astral tree of the nuclear genes, and in Kikihia, the various Kikihia species generally arrange themselves into clades corresponding to habitat types. So for instance, you have a green foliage or forest clade, you have a dry scrub clade, and you have a grassland and shrubland clade. However, when you look at the mitochondrial tree, you see that there is a clade that's only in the mitochondrial tree that has species from all 3 different habitat types. You have some species that are dry scrub, some species that are grassland, and some species that are forest. And the potential explanation for this mitochondrial clade that is not found in the nuclear phylogenomic tree is that it formed through multiple rounds of hybridization. So multiple rounds of hybridization occurred that passed mitochondrion from species to species. Similarly, when I look at the Maoricicada nuclear phylogenomic and mitochondrial genome trees, I find that there are also mitochondrial nuclear discordance. Similar to Kikihia, the Maoricicada tree shows species dividing themselves up into roughly ecological clades, where I have a group of lowland cicadas down here, and then a large alpine radiation, which includes some subalpine species. And there are several instances of mitonuclear discordance in this data set that could be explained through hybridization. First of all, you have the lowland species Maoricicada hamiltoni, which in the nuclear tree is sister to all other Maoricicada, but in the mitochondrial tree groups with other lowland Maoricicada, and so that could be the result of hybridization, potentially shown here with this arrow. You also have the low elevation species Maoricicada iolanthe and the low to mid elevation species Maoricicada campbelli, which in the nuclear tree group with the other lowland species, while in the mitochondrial tree group within the alpine radiation. So that could be the result of hybridization with an alpine lineage. And finally, you have a population of the subalpine species Maoricicada mangu that in the nuclear tree groups with the other members of its other populations in its species, but in the mitochondrial tree groups that with not particularly well support elsewhere in the alpine radiation, which it could be explained by hybridization with an unsampled, possibly extinct ghost lineage. So to further investigate these potential hybridization events, I turn to phylogenetic network methods. And so for this talk, I will be looking at two phylogenetic network methods, SNAC and phyNest. Both of these are composite likelihood methods, but they differ in the data that they use as input. So SNAC is a composite likelihood method that uses the gene tree topologies as input, while phyNest uses the actual sequence data as input. But both methods have computational limitations, which mean that for large data sets, I've found you need to subset the taxa into a smaller data set in order for these methods to run properly or efficiently. So in brief, going over my methods, I used the same phylogenomic data set that I had used for generating my mitonuclear discordance results that I introduced earlier. So this is an anchored hybrid enrichment data set of around 500 nuclear genes, along with recovered mitochondrial genomes. I used Mr. Bayes gene trees as input for SNAC following the ticker pipeline, which I will not get into in this talk. And I used a concatenated sequence alignment as input for phyNest. And for both methods, I subset the taxa for all the analyses for computational tractability, focusing on particular hypotheses. So moving on to my hypotheses, starting with Kikihia, I first have a hypothesis about the formation of this weird mitochondrial clade containing species from 3 different habitat types that for the rest of this talk I will call the Westlandica group. And my hypothesis is that this mitochondrial clade containing forest edge, grass shrub, and dry scrub cicadas is the result of multiple rounds of hybridization. Second, I have a hypothesis on the origin of 2 separate species, Kikihia angusta and Kikihia and Maoricicada. My hypothesis is that the angusta, which is a grass cicada, and Kikihia and Maoricicada, which is a dry scrub cicada, are related to cicadas of the respected habitat types and that they hybridized with each other. We know that they hybridized with each other, but what are their close relatives without hybridization is unclear. And so I'm hypothesized that they are related to other cicadas from their respective habitat types. And finally, previous researchers have identified a population of the species Kikihia muta that has mitochondrial DNA of a different undescribed species, Kikihia tuta. And I hypothesize that phylogenetic network methods will uncover a hybridization event between Kikihia muta and Kikihia tuta, forming this muta tuta population. So because of the limitations for taxon sets, I could not run SNAC on all of my Kikihia species at once. So I decided to split the Kikihia data set into 3 different subsets. each one focusing on densely sampling one of those major ecological-based clades while sparsely sampling all the others. So the first subset densely samples the green foliage and forest cicadas, while sparsely sampling the other clades. The second subset densely samples the dry scrub clade, and the third subset densely samples the grass and shrub clade, while also including all the cicadas from that potentially artifact from that weird mitochondrial Westlandica group. So moving on to the results, first looking at that first taxon set, densely sampling the green foliage clade, I first find a couple of hybridization events among the various subspecies and populations within the Kikihia cutora species complex, which is a widely distributed green foliage cicada species complex, including some hybridization events between individuals found on the North Island and populations and subspecies that are found on offshore islands like Norfolk Island or the Kermadex. I also find a hybridization event between two grass cicadas, Kikihia muta and the undescribed Kikihia tasmani, which interestingly do not currently encounter each other. Their species distributions do not overlap or are not adjacent. So it must mean that either a hybridization event occurred in the past when they had greater species distributions, or the hybridization event was actually occurred through a third-party species lineage that is not sampled in this subset. And finally, I find a hybridization event connecting the forest edge Westlandica group species Kikihia subalpina and Flemingi with a collection of grass and dry scrub cicadas found across the South Island, which I interpret as potential evidence for the Westlandica group hypothesis. Moving on to the second taxon set, which densely samples the dry scrub cicadas, I find a couple of hybridization events among members of this clade, these dry scrub cicadas, which, if you notice on the map, do not have overlapping or adjacent species ranges, which indicates that when the hybridization events occurred, they were probably happened when these species had larger species ranges, so they were able to encounter each other. That's the most reasonable possibility to me. And I also find the same hybridization event between the Westlandica group species subalpina and Flemingi and the South Island grass and dry scrub cicadas, which again is support for the Westlandica group hypothesis. Moving on to the third taxon set, the which densely samples the grass and shrub cicadas. I find a hybridization event between some of the cicadas that are generally considered part of this artifactual Westlandica group, namely the dry scrub cicada Kikihia and Maoricicada, which is in this dotted species range here, with the grass cicadas Tasmani, Westlandica North, and Westlandica South. So I interpret this as evidence for the Westlandica group hypothesis. Interestingly, this subset does not show any hybridization events between subalpina and Flemingi and the other Westlandica group members, which is unusual because the previous two taxon subsets did show that hybridization event. I also find a hybridization event inferred between the species Kikihia tuta and the muta tuta population, albeit in the wrong direction, which is evidence for the muta tuta hybridization hypothesis. However, I have found that phylogenetic network methods apparently do sometimes infer hybridization edges in the wrong direction, particularly SNAC, because it is a method that infers unrooted networks. And I also find hybridization involving the grass cicada Kikihia angusta and the dry scrub cicada Kikihia rosea, which is evidence, which is evidence that doesn't support my hypothesis for the origin of Kikihia, angusta, and Maoricicada, because Kikihia angusta, I hypothesize that Kikihia angusta is more closely related to grass cicadas than it is to dry scrub cicadas. So moving on to my FINEST results, I found that for FINEST, this program is much more restrictive in the number of taxa than SNAC, meaning I could not use the same taxon subsets that I had used in my SNAC runs. So instead, I made new taxon sets that focus on specific hybridization hypotheses. So I made a taxon set that focuses on Kikihia angusta and Maoricicada, and some grass and dry scrub cicadas that are potential relatives. I made a taxon set focusing on Mutatuda and its potential relatives, and I made a couple of subsets focusing on the species in this artifactual Westlandica group. So looking at the Angusta and Maoricicada set, I again find hybridization between Angusta and Rosia, which, as I mentioned before, does not really support my hypothesis for the origin of Kikihia angusta. I find hybridization between Kikihia muta, Kikihia tuta, and Kikihia mutatuta, which in this second taxon set, which is evidence for the hybrid origin of Mutatuda, with the added bonus that the species Kikihia paxilluli is also involved. And looking at my first taxon set with the Westlandica group species, I find a hybridization event between the Westlandica group species Maoricicada and Westlandica south. And I find when I add in Flemmingi and subalpina, I find a hybridization event connecting Flemmingi and subalpina with the broader South Island Westlandica group species, which is evidence for the Westlandica group hypothesis. So to conclude, and the conclusions that I've drawn from my phylogenetic network analyses for Kikihia are that the Westlandica group origin hypothesis is partially supported. I found that subalpina and Flemmingi hybridized with an ancestor of most South Island Kikihia. I find that Kikihia mutatuta is recovered as a hybrid between Kikihia muta and Kikihia tuta. But I find that the origin of Kikihia angusta and Maoricicada remains unclear. And I find that across both SNAC and FINEST, the results appear to be quite sensitive to taxon sampling, which is important because for both methods, I cannot use all the species in my data set in a single run. So the fact that I have to subset my taxa down means that my results may change depending on how I choose to subset my taxa, making interpretation somewhat more difficult. Moving up for the rest of the talk, I will be talking about my Maoricicada results. And so I will start off with my Maoricicada hypotheses. First off, I hypothesized that the lowland cicada Hamiltoni, which I have mentioned earlier, hybridized with the other lowland Maoricicada. I hypothesized that there is alpine introgression or hybridization into the lowland species Iolanthe and low to mid elevation Campbelli. I hypothesized there will be evidence for ghost introgression into the this unusual population of Maoricicada mangu. And I hypothesized that some individuals of the alpine species Maoricicada oromelena, which when they were collected were originally thought to be hybrids, will be identified as hybrids between oromelena and some other alpine species, namely Maoricicada clamitans and Maoricicada cassiope. So first, looking at the SNAC results for Maoricicada, because there are fewer species in Maoricicada than in Kikihia, I did not need to subsample my taxa for the Maoricicada SNAC analyses. All I needed to do was reduce the number of individuals for each taxon, for each species. So first of all, I find a hybridization event between the or among the alpine species Maoricicada nigra Maoricicada phaeoptera, and otagoensis, which, as you can see on these maps, have overlapping species distributions, which supports the idea that they could have potentially hybridized with each other. I find that SNAC recovers a hybridization event between the alpine species Maoricicada climatans and the unusual Maoricicada cassiamolans individual. That was that potential hybrid Maoricicada oromelena individual. So that's evidence for the cassiamolans being being a hybrid. And while I do not find evidence of hybridization between alpine species and the lowland species Campbelli and Iolanthe, I do find hybridization between the lowland species Iolanthe and Meyersi, which is interesting. In order to investigate these results further, I did decide to subsample my taxa for the Maoricicada dataset, but this time including all of the individuals for the alpine species and the lowland species to see if there's interesting things between different populations of the same species. And so when I look at all of the individuals for the alpine Maoricicada, I find that the hybridization event between climatans and oromelena and cassiamolans is continues to be recovered, which is evidence that the unusual phenotype of the cassiamolans individuals is due to hybridization. I also, when I look at the lowland subset with all individuals, I once again do not find any evidence of Maoricicada campbelli and Iolanthe having alpine hybridization, but I do find that hybridization between the lowland species Maoricicada and Iolanthe. Notably, I do not find any hybridization between the Hamiltoni species and the other lowland species, like I hypothesized. So to conclude for my Maoricicada hypotheses, I was not able to present any finest results for Maoricicada because I'm still troubleshooting those analyses. I'm finding that they are quite difficult right now. But to conclude my hypotheses, I find no evidence of Maoricicada Hamiltoni hybridization. I find no evidence of alpine integration or hybridization into Campbelli and Iolanthe, but I do find evidence of hybridization between Iolanthe and the fellow lowland species Meyersi. I find no evidence of hybridization involving this population of Maoricicada mangu, the Maoricicada mangu awakino. But I do find hybridization between the alpine species M. climatans and M. oromelena, which explains those unusual cassiamolans individuals. So for some future steps, I would like to finish troubleshooting and running the Maoricicada finest analyses and see how they compare with my SNAC results. And I would also like to expand my work to compare with additional network methods, such as various methods included in the software package phylogenetic. So I'd like to acknowledge the Simon Lab at the University of Connecticut and previous New Zealand cicada researchers affiliated. I'd like to acknowledge computational research resources at the University of Connecticut that I've used. I'd like to acknowledge the Fulbright New Zealand for bringing me to New Zealand, and I'd like to thank the University of Auckland and University of Auckland CCE for hosting me. And with that, I will take any questions.